So when we left off, we had gone from a single diploid cell to 2 haploid cells. And so remember that this diploid cell is 2n, right? So each chromosome has a pair. This big chromosome is paired with this big chromosome. This small chromosome is paired with this small chromosome. And after the first few steps of meiosis, each of these cells is now haploid, right? So those pairs have been split up, right? A big chromosome here and a small chromosome, a big chromosome and a small chromosome, but they only split up after they had done some shuffling, right? Some rearranging. And this part of meiosis, this set of steps, we call meiosis I. However, recall that in the context of the cell cycle, this is S phase. This is after S phase. So instead of going into M phase, the progenitor cell went into meiosis I. And so there are still 2 copies of each gene in each of these cells, right? So let's say that our eye color gene is being carried here. That eye color gene is also there. And we want gametes, we want haploid cells for reproduction that only have 1 copy of each gene. And so what that means is that each of these haploid cells has to split again. And this process looks really, really similar to mitosis. And so I'm just going to follow this cell right here through the next set of steps. And so now we move into prophase 2, where the cytoskeleton begins to the spindle apparatus begins to form. I'll go ahead and keep drawing our chromosomes here. And then, after prophase 2, we get metaphase 2. And as in metaphase of mitosis, now those chromosomes are going to line up down the center of the cell in the metaphase plate. And again, our spindle apparatus is assembling. It's attaching microtubules to each of the chromatids. And now the kinetochore that is holding those chromatids together disassembles. And those individual chromatids begin moving to opposite sides of the cell. Right, and so now we've got just 1 chromatid over here for each of our chromosomes, and the other chromatid headed over here. And just like in mitosis, this is called anaphase. But because this is the second half of mitosis, this is anaphase 2. And then, of course, once anaphase has happened, then the nuclear envelopes reform, and you end up with 1 fully formed gamete in a single cell, and another fully formed gamete in a single cell. And this is telophase 2 and cytokinesis. So remember, for this part of my drawing, I'm only following a single one of these haploid cells. And so this haploid cell is doing this as well. And where we started with one cell that was diploid and duplicated, now we end up with 1, 2, and then the 3 and the 4 cells that this intermediate cell produces, which are haploid and not duplicated. Okay, so this seems like a lot of work, right? And there are lots of possible ways to fail. Why do organisms go to all of this trouble? Well, that's our next topic.